Hello, everybody. My name is Carlson. I am a new PhD student at the Hawkeye in Professor Rosenbaum's lab. And today, I will be presenting to you my work on what I affectionately like to call electron taxis. Now, before I get to that, I would like to introduce you guys to somebody very special. Meet E. coli. Say hi, E. coli. Hello. Now, E. coli here is the workhorse of biology. Look at those muscles. Look at that flex. Immaculate. So we, as biologists, we like to make sure to keep E. coli very well fed. E. coli, would you like some food? That's a yes. Let's give our buddy here some food. Some Jena. We're in Germany, we give the E. coli Jena. It likes it. So, anyway, when the food gets into the cell, electrons are freed from the food. See, they're a very energetic bunch, right? Now, the cell, it wants to capture that energy and convert it into something that it can use. In order to do that, it sort of has like a pathway. And as the electrons travel this pathway, their energy is converted into a molecule called ATP. ATP, you can think of like the gasoline for the cell. Now, the E. coli uses this ATP to make us things. This is what makes them the workhorse. So in this case, it made us some insulin. Diabetes be gone. Yes. Now, besides food, it's also very important that you provide plenty of oxygen for the cell. See, oxygen picks up the electrons at the end of their journey. Just wait for it. And then releases them from the cell back into the environment. Now, this is very important. If there is not oxygen at the end of their journey to pick them up and free them from the cell, the electrons, they have to find somewhere else to go. They have to find another exit, and the cell misses out on some of that energy. Worst case scenario, there's not enough exits for how many electrons there are. And the electrons, they build and they build until the cell dies. So, we would like to avoid that. But there's a bit of another catch. What if we have a nice natural product like this guy here, but they're insensitive to oxygen? What are we going to do? Well, that's where bioelectrochemical systems comes in. Now, you might ask yourself, what is a bioelectrochemical system? Well, it sounds complicated, but it's actually really quite simple. It is the interaction between biology and electrodes. Usually it's not this dramatic, it's more. Well, yes. So, we plan on using this to solve our oxygen issue. Now, the solution begins with a class of chemicals that we call mediators. Now, a mediator is basically, very simply put, an electron taxi. It can pick up electrons in the cell and carry them somewhere, like an electrode. So let's see that in action. So the taxis come in and pick up the electrons from their pathway. And then, unlike oxygen, which can just, go out into the environment and the electrons are gone, the taxis need somewhere to drop them off. So they drop off their electrons at the electrode. This establishes our bioelectrochemical system. And then it can use the ATP generated to produce us oxygen-sensitive natural products. So this is what I do. I go in and I engineer the cell to make its own taxis. And then I am tuning it to sort of make this process a little better. Make sure that they're producing enough ATP for them to grow, thrive, and more importantly, work. And so, in summary, I genetically engineered E. coli to produce their own mediators so that we can establish a bioelectrochemical system that circumvents their oxygen needs and allows us to produce natural products that are destroyed by oxygen. Thank you, and I would like to thank Dr. Angel Franco, who was my supervisor when I began working on this topic. I would also like to thank the European Research Council for their generous funding through the eMicro Consolidator grant, and finally I would like to thank the entire biotechnology team at the Hawkeye, on which I am a part of. Thank you very much.
Suga po.